Okay, thank you very much for the opportunity to present your favorite nanogels here in the symposium. I'm enjoying it a lot, and I hope you will also enjoy my presentation now. And this is about the use of thermoresponsive nanogels for topical drug delivery, most specifically to deliver into the skin. Just to give a brief overview to all of you, an introduction about our working place. This is Polymat. This is a research institution that is dealing with the research and development of different polymers here in the Basque country in Spain. We are located in the northeast of Spain, right next to the limit with France. And our institution is divided in two kind of lines of research. One of them more devoted to the science of from the university point of view, and another one also related more to applications with industries. But well, this place where we are actually working, San Sebastian, is a very fascinating and beautiful place, in case some of you know it, and you already know this. But then also, just I wanted to highlight an article that it was published in the editorial in 2019 in ACS Nano, where they mentioned that San Sebastian is the city of nanoscience and technology. And I think I can really say that. I moved here almost 4 years ago, and I'm really enjoying all this vivid environment, scientific environment that we have here in San Sebastian. You can see we have located in one of the campus of the University of the Basque country. But then here we are able to collaborate with a huge variety of expertises, ranging from nanotechnology to physics to some technical companies also working in the field, and also, for instance, a research institution linked to the hospital. It's really fascinating here. And about the work we do, we are recently joined Group 2 Polymat. Now, as I said, almost 4 years. For me, this is a short time. No, I think several of you will agree to this. And here we join, actually my group joined after spending almost 15 years in Germany, in Berlin. And then we established here a research line dealing with drug delivery applications. So we specialize ourselves on the use of responsive polymers. And then later also we got 2 research lines, independent research lines, that joined me to work on the synthesis of polymers around enzymes and to improve the properties of these enzymes, finding applications in biocatalysis and biomedicine. And this is led by Anna Bellocchi. And then we have another research line that joined later that is dealing with the development of smart biomaterials for tissue engineering. And this is led by Dr. Sandra Camarero. But well, specifically about the talk today, I will be showing some of the basics that we should all know to understand the results that I will show later about dermal drug delivery. I will talk a little bit about the skin barrier function and some of the biochemical triggers that it can be used to induce a smart drug delivery on this sort of applications. And I will illustrate this with some of the results of our group regarding the synthesis of nanogels and their evaluation regarding skin penetration and also skin drug delivery. So just to get started, I think all of you already know that the skin is our major organ in our body and is responsible for isolating us, so to say, from the environment. They are really specialized on working as a barrier, and therefore they are really difficult when one wants to deliver some molecules across them. Even though that we have the chances to deliver some small molecules and big molecules, the big molecules are actually hindered, and this is why a research group like ours are working on the development of new concepts to deliver big molecules inside of the skin. And most of the molecules that one tried to deliver inside of the skin are being rejected or somehow filtered by this the first layer of the stratum corneum, that is this one with dead cells, the so-called stratum corneum. But then beneath this first layer, we have a second layer of viable cells. No? Both together they comprise the epidermis, but these 2 layers are the outermost layers of the skin. But one could say that one part of them is dead cells. We got a lot of noise, but from Tuana, wait. Somebody can you please? Yeah, thank you. And then beneath this first layer of the epidermis, then we have also dermis, where the cellular complexity increases. And then one can also find blood vessels there and other type of cells. What is interesting on the skin is that we also have the opportunity to deliver into the hair follicle. And this is particularly interesting for big molecules like the nanocarriers. And since here we have a relatively bigger hole or pores where our materials could go, but of course, some engineering of the material is needed to achieve this. Regarding the opportunities for drug delivery into the skin, there are several of them. Of course, one of them that is very, very popular is the one regarding cosmetics. Yeah, here I just illustrated with anti-aging, but there are many approaches to develop systems for cosmetics. And then, of course, we have one that is a very common disease, acne, and acne treatment is particularly important when we want to develop systems for the delivery into the hair follicle. Very widely spread diseases like psoriasis and atopic dermatitis are also interesting targets for drug delivery applications. And there are also several diseases that are rare diseases. And one of them that I would like to highlight today is the so-called autosomal recessive congenital ichthyosis. That is a monogenic disease that, to which I will come a little bit later with a little bit more of details. But there are several of diseases like this one that are rare and have nowadays no treatment at all. Regarding the strategies that are currently used for the delivery of molecules inside of the skin, one could classify them in two big groups, no? Like some of them that are using penetration enhancement methodologies, and another one using particulated delivery systems. And regarding the penetration enhancement, here we could have physical or chemical enhancements. No? Just as an example, yesterday we saw a fantastic talk where they use microneedles as a physical penetration enhancement methodology. And then in the case, for instance, with direct delivery systems, we also saw recently, just a couple of, one or two talks before mine, we could see some examples where hydrogels are being used to enhance the delivery of materials into the skin, and also amphibites. No? And the most classical ones when we talk about nanoparticulated systems, we will have the polymeric nanoparticles, of course, the solid lipid nanoparticles, very, very popular. And here we can also have as a group the nanocrystals, which are more and more coming into play. And regarding the properties of these materials, regarding mechanical properties, sizes, stability, etc., the materials that we develop, which are these cross-link polymers, the nanogels, are in between, I would say, between polymeric nanoparticles and solid lipid nanoparticles. Yeah, and yes, okay, this is what we hypothesize in our research, is that these systems will have a potential to somehow overcome the issues of these already established technologies in the drug delivery field in the skin. So then here we really postulated several hypotheses, and we had the privilege to work with several researchers, cosmetologists, dermatologists, etc., when we were in Berlin, and we could really somehow target several hypotheses. But today I will just only go into a couple of them. But these were related to the degradability of the material, to the size, for instance, analyzing how the size influences the penetration of the particles on some of the layers of the skin or on the hair follicle, for instance, formulation strategies, for instance, and then also responsive modalities, etc. But well, one of the hypotheses, or several of these hypotheses, are actually summarized in the review that we recently published. This was published this year in Angewandte Chemie. Where we explore, where we do a very nice comprehensive study of the nanocarriers that are being developed nowadays for the skin applications. And I invite all of you to have a look into this review, because I think it's a really nice review of the state of the art in the field. But well, regarding the activities in our group, we have explored several of them. One, for instance, that I will not discuss today, but I would like to anyway mention, is the development of nanogels as vaccines, and we have some work done in this area. Well, and there are some others that I will try to highlight throughout the presentation today. Regarding the treatment modality that we propose here, is one of the hypotheses is to use these thermoresponsive materials that the polymeric community here knows very, very well. And for instance, NIPAM and isopropyl acrylamide has been used and studied a lot for several decades. So we know that this polymer has a transition of around 32, 33 degrees approximately, depending on the methodology of synthesis and copolymers we use. And it happened that these sort of polymers are especially interesting for drug delivery applications, because on the skin we have actually natural thermal gradient. Yeah, so when we have very much on the surface of the skin, the temperature is around 32 degrees. And when we go inside of the first layer of the skin, this temperature increases for several degrees and until we reach 37. So we have here really nice window on changes on temperature that it could be used to trigger changes on our thermoresponsive polymers. And this is why we were interested in using these thermoresponsive materials on this application. Of course, we have the chance also to heat up a little bit the skin. When I mean a little bit, is just only to 40 degrees approximately. And this is a typical approach that is done with an infrared lamp. And we have also explored that. And a field that we are nowadays exploring a little bit more, but we are still studying this, is exploring some of the proteins or any other kind of biological signals that it could also be used to somehow induce changes on the properties of the polymer, like degradation, swelling, etc., etc. So in our study, trying to target several of these hypotheses that we initially had, we somehow postulated our research in three main areas. So one of them focused on the synthesis and the preliminary evaluation of their cytocompatibility or biocompatibility of the materials. We also postulated the analysis of the different mechanisms by which our polymers may or may not penetrate on the skin, and also they may or may not induce the penetration of some cargo molecules inside of them. And then also at the end, we have also explored actual applications. No? So we analyzed what was the therapeutic outcome of these approaches we had. So jumping into the first one. So here, since we wanted to work with thermoresponsive materials, and we wanted to somehow confine the size of two diameters around 100 to 200 nanometers, we got excited on working with nanogels or these cross-linking materials, where we use as cross-linking point different sort of polymers or small cross-linkers. Here I'm illustrating, for instance, dendritic polyglycerol or hyperrange polyglycerol as a cross-linker, but we have also used bisacrylamide, for instance, and another popular cross-linkers. Also, we use actually inorganic materials as cross-linker to also induce some other sort of responses on the systems. But just focus on the temperature here. What we pursue here, it was to have a response on the nanogel, going from a swollen system, like imagine having a sponge completely soaked with water or with molecules. And then when we increase the temperature of this, we press this sponge, and the sponge releases the molecules that are inside. And these molecules being the cargo molecules we have, but also what is actually very important, being water. No? So we should also keep this in mind, that our material will release water. And this is especially important on topical drug delivery, because hydration of the skin is also one methodology of penetration enhancement. Regarding the thermoresponsive materials we use, we screen a huge family of popular, the usual suspects, so to say. And we had a huge team of people working on this for almost 10 years, developing, fine-tuning the sizes, fine-tuning the transition temperatures, etc., etc. And one example of the work that we typically do is the work that Loreen Toyne did when she was doing the PhD with me in Berlin. It's with copolymerize, for instance, an acrylated hyperrange polymer. We copolymerize it with NIPAM or with NIPMAM. That is a very analog monomer that has a transition temperature around 48 degrees. And then by playing with the conditions of the polymerization, concentrations, concentration of surfactant use or not of catalyst, etc., we could then control the properties of our nanogels. No? And some of the examples of the property we could control, on one side, we can see here that we can control the sizes, no? By changing the acrylation degree of the cross-linker. And we can also change, for instance, the temperature of transition and the sizes when we change the ratio between the copolymers that we use, or the comonomers that we use. So here we can see that when we move only 100% NIPAM, we have a transition temperature of around 34, 35 degrees. And then when we start copolymerizing with NIPMAM, this increases. And in the case we have a 100% NIPMAM copolymerized with a cross-linker, we have a transition temperature around 46 degrees. So we have here just a range of materials that eventually will enable us to test all the three, or at least two chances of triggering with temperature that we have, either the natural gradient of the skin or irradiation with infrared lamp. Another methodology that we use for the synthesis, it was the nanoprecipitation. Here we pre-synthesize the cross-linker, and then we also pre-synthesize the thermoresponsive polymer. So here we have a better control on the length of the thermoresponsive segment. We functionalize at the end. We make these telecalic polymers, and then we play with the nanoprecipitation here. Now, again, we have to play with concentrations, injection speed, striding speed, et cetera, But then we come from injection and a solvent, where we had all our polymers or precursors completely dissolved into a non-solvent. This will generate the nucleation for the polymerization, and then ultimately will give us the nanogel as a product. So here it was very interesting because initially we used a solvent DMF as a non-solvent water, warm water, so to say. But then we started also changing this strategy, for instance, to use just cold water as a solvent because these are thermoresponsive segments, and then warm water as a non-solvent, and then we just did a thermonanoprecipitation water in water. And of course, here we could as well control sizes, functionalities, etc. Just to finish this first part, before we move to actual biological assays, we have to demonstrate that these materials are biocompatible, at least on preliminary studies. And in collaboration with the Cloister Lab, that was in University Potsdam back then, we ran a huge set of experiments analyzing the typical studies of cell viability, cell cytotoxicity. We analyze as well the retention potential of the materials. We analyze if they could exert oxidative stress or any other type of toxicity, like for instance, genotoxicity. And in all cases, comparing them against positive and negative controls, we could see that our materials actually have a very interesting biocompatible profile. So this was highly encouraging. And then therefore we took these materials into more advanced models. And in these advanced models, we tried to understand what was the mechanism of action of these materials, or the potential of action of these materials in the skin. And here, in collaboration with the group at the Charité Hospital, in this case was with Ferenza Rankan. She's a specialist working with excised human skin. Here, we first analyzed how the penetration of the nanogels occurs on the skin, and how the penetration of eventual cargo of the nanogel will penetrate in the skin. And what we could see here is when we have our nanogel fluorescently labeled, I just take it to this image just to make it simple, the analysis. We could see that our nanogels, they mostly accumulate on the outermost part of the skin, so in the stratum corneum, on this layer of dead cells. And then when we see how the cargo molecule penetrates, we could see that the cargo molecule actually goes beyond this stratum corneum, and it could penetrate into the first layers of the viable epidermis. But we could also very nicely see that they penetrate on the dermis, which is in most of the case very, very difficult. We quantify this. We could correlate this with some of the properties of the nanogels. Sorry that we're not going into the details on that because of the time. But what I can tell you is that in principle, we could see here very nicely that by changing or fine-tuning some properties of the nanogels, we could enhance the penetration of the cargo molecule inside of the skin. But in all cases, the nanogel gets stuck in the stratum corneum, which is interesting to see, of course, and it's somehow an obvious result. We as well analyze what happened with the skin when we incubated the skin with our nanogels and our nanogel solutions. And we could see that the skin very, very quickly hydrates, no? And as I mentioned before, this expelling of the water when we change when we cross the transition temperature with the nanogels is something that is interesting to have on these sort of systems, because as I said before, the water is known to be also a penetration enhancer, a natural penetration enhancer of the skin. And actually we could see that our nanogels very, very quickly hydrate the skin. And also what it was interesting to see is that also they deorganize the packing between lipids and proteins in the skin. Following this hypothesis, we compare systems with different architectures. For instance, in this case, we moved to work with nanogels, but now with hollow core, so sort of capsules. We compare them with a hard core, and then we compare the penetration of just water into the skin. And we analyze this by Raman, and we could very nicely see that in those cases, we have our nanocapsules or dash nanogels. We could see that they actually really increase the penetration of water in a very short time. And this penetration of water, it seems to be enhanced at the moment we increase the temperature of the skin. No? So actually this hypothesis we had of the nanogels squeezing and releasing some water, it seems that also has an effect on the hydration capacity of the nanogels of the skin. And this is actually very interesting result. And just to complete the analysis of the different potential mechanisms for drug delivery of our nanogels, together with Fitz and Sale, as well from Charité, we analyze how these nanogels may or could penetrate inside of the hair follicles. And here we analyze this penetration on porcine ears. And what Fitzon could very nicely see here is that our nanogels actually has a penetration that is dependent on the size of the nanogels, or the diameter they have in solution. As it was already reported for other polymers, it seems that when the polymer has diameters of around 500 nanometers, they have an optimal penetration on the hair follicles. And this is what we could see here when we compare the penetration of the nanogels into the hair follicles. And then this was also correlated with the penetration of the cargo molecule that was inside. No? So nowadays, we are also interested in understanding how this actually happened. And we are also interested in understanding how the other properties may play a role on the penetration of the hair follicle. And one of them will be the mechanical property. And this is basically the subject of the Marie Curie fellow that is now in my group. But well, moving a bit forward, just to take advantage of the few minutes I still have remaining, I would like to show you some of the potential applications that these materials may have. No? Since nanogels are relatively big nanocarriers in terms of the cargo molecules that they could have. So we analyze here also the usual suspects, what we could encapsulate within the systems, no? Like dexamethasone, doxorubicin, diclofenac, methotrexate, etc. All these small molecules. And we could see that this, always working with these small molecules, we had a release of the molecules from the nanogels because of diffusion. No? And this is kind of understandable, thinking that the pore size of the nanogels is not probably small enough to really conceal the molecules to the inner space of the nanogels. So this on-off release that we are hoping to have, it seems that with small molecules does not really work well. But when we move to bigger molecules and cargos like asparaginase, and even so big ones like etanercept, that is a fusion protein with a molecular weight of 150 kilodalton, we could see that these nanogels are really good on encapsulating them with an encapsulation efficiency that is very, very high and a loading capacity of almost 100% compared to the weight of the nanogel. And then we could have a very nice control on the release that is mostly occurring when we increase the temperature and we cross through the transition temperature. And particularly today is a molecule that is very interesting to consider is this molecule here, this transglutaminase, because it's going to be the subject of the next experiments. Before we move to these expensive proteins, and we always work with albumin as a model, no? Specifically FITC labeled albumin, because then we can track it very nicely inside of the skin and also on the experiments in the lab. But here we wanted to prove this concept I mentioned before. What happens if we have nanogels that have the transition temperature set to release the protein only in the cases we face this natural thermal gradient? And what happens if we have another nanogel where the temperature of transition is set to just only release the protein with a heat source, namely the infrared lamp? And in this case, we analyze how the encapsulation and release of the albumin as a model here fits the label albumin. And in the case we work with our nanogels with NIPAM, just as a reminder, it has a transition temperature around 34, 35 degrees. We could see that only on those cases that we work with this on the temperatures that cross the transition temperature, we have a very quantitative release of the protein, but when we are below the transition temperature, this release is hindered, and it's pretty much what we expected to see. When we work with the copolymer 50-50, we have a transition around 40 degrees. So now when we incubate this at 29 and 37, the release is not that prominent as is compared whenever we cross the transition temperature at 42 degrees. And when our nanogel is now having a transition temperature that is much higher, so when we incubate at these two temperatures, then the proteins are almost not having a very marginal release open this very long window of time. So they're somehow encapsulating the proteins and releasing them with the behavior we were expecting. And now in collaboration with Sarah Hetrich from the Freie Universität Berlin at that time, then later in British Columbia University in Canada, we could demonstrate very nicely that these results also correlate with the delivery capacity of the materials inside of the skin, skin models and human skin. In this case, we analyzed the penetration of the proteins and also excised human skin, and we could see that only when we have this natural temperature gradient on the skin, we will have a nice penetration of the FITC label albumin in stratum corneum and viable epidermis, only when they were encapsulated with the NIPAM nanogels, pretty much the same of what we saw before. And then when we irradiated with an infrared lamp, now we have not only the penetration of the protein that it was encapsulated on the NIPAM nanogels, but also we have penetration protein from the that one they were encapsulated on the copolymer NIPAM nanogels. So that is pretty much what we could see in the lab. We could also reproduce on the ex vivo experiments, which was really encouraging. And then just to show you a couple of more slides on the application or actual application of this, here for the treatment of the autosomal recessive congenital ichthyosis, delivering transglutaminase will be one of the approaches to treat this disease, this rare disease. And then by this delivery of the transglutaminase, what we expect is to recover the barrier function of the skin. And this is pretty much what we could see on reconstructed skin models from Sarah as well. And then we could also see that whenever the disease it was present, it was a high penetration of a model molecule, but whenever we deliver transglutaminase, there is no penetration anymore, and the skin recovered the barrier function. And this was on just a simple model, but then we also did this analysis on a very complex model using cells from patients, and they were also constructed on a skin equivalence in Sarah's group. And here we could demonstrate that exactly happens the same on this more therapeutically relevant model. So we recover the barrier function of the skin upon the delivery of transglutaminase. That is mostly occurring when we deliver the transglutaminase with our nanogels as compared to the nanogels to the transglutaminase delivered in the solution. And this is where we are also working nowadays, but to deliver other type of molecules, but for the same disease. And then coming to the end, very briefly to summarize that we established these synthetic methodologies, and we could show that the materials are quite have a strong potential for the delivery regarding the biocompatible profile they have, and also the different mechanisms of delivery they may have in the skin. We transfer most of this knowledge to other sort of diseases. In the line of what I showed today, we keep on working on cisgenodermatosis, but also now we move to work in alopecia areata, and we got funding from two sources to do that. But with this, I come to the end to acknowledge the people that are still working on this in the group, and to acknowledge all of you, of course, for your interest and for listening to me today. Thank you very much.
Thank you, Marcelo. Very